All right, this is a video of the solutions for genetics problems 1 through 11, our first homework set. So the first two questions are really making sure you understand the vocabulary. So what does homozygous mean and what does heterozygous mean? So problem number 1 says to underline the genotypes that are homozygous for all traits. So that would include 2, 5, 7, 9, and 10. Problem 2 asks us to circle the genotypes that are heterozygous for all traits. That would be 3, 4, and 11. So now let's talk about some problems. In man, normal pigmentation is due to a dominant gene, and albinism, no pigmentation to its recessive allele. The gene is autosomal, which means it's not carried on a sex chromosome. So that means we will have no X's and Y's in our Punnett Square. So problem number 3, a normal man marries an albino woman. Their first child is albino. So the mother is a because she is albino. Their first child is albino, which means the father had to have been a carrier for albinism. So we put the gametes go on the outside, they would be haploid, and the possible offspring go inside the Punnett Square. So the chances that future children would be albino would be 50% or 1/2, and non-albino would be 50% or 1/2. The reason that you know that the father has to be heterozygous is because if they have a child who is homozygous recessive, that child got one allele from mother, but that child also had to have gotten another allele from the father. So that means the father must have the recessive allele in his cells, although he doesn't express the trait because he has the dominant allele for normal pigmentation. Problem 4, an albino man marries a normally pigmented woman. They have 9 children that are all normally pigmented. What is the probable genotype of the father? The father is albino, so he is a. The mother is likely big A because they have 9 children that are normally pigmented. She could be heterozygous, but it would be very unlikely. And the children would all be heterozygous. So here are our possible Punnett squares. This one is more likely. And this would be considered a monohybrid test cross. Well, not really, because you cannot do test crosses with humans. A test cross is when you have an individual that has a dominant phenotype and you don't know whether they are heterozygous for that dominant phenotype or homozygous dominant. And so you would cross them with an individual who's homozygous recessive. And then if any of their offspring have the recessive phenotype, you know they were heterozygous. You can do this with guinea pigs or fruit flies, but not really so much with humans. But you could kind of consider this a test cross. A normally pigmented man whose father was an albino marries an albino woman whose parents were both normal. So let's break this down. You have a normally pigmented man whose father was an albino. So the father's father is a because he's albino. The father is heterozygous, and you know that because if his father was an albino, the only allele that his father could have passed down to him would be the recessive allele. But he's not albino, so he is heterozygous. So he marries an albino woman whose parents were both normal. So the mother's mother must have been heterozygous if she passed down the albinism trait to her daughter, and the mother is a. So we're putting this father and this mother in our Punnett Square. And you have a 50% chance of getting normal children and a 50% chance of getting albino children. Now, generally speaking, we put the male's gametes across the top and the female's gametes down the side, but we didn't do that in this particular Punnett square. It doesn't really matter because you get the same probability. You just would have different locations, but the numbers would be the same. So don't really worry about that so much. So we have a heterozygous rough-coated guinea pig, and that heterozygous rough-coated guinea pig is mated with a rough-coated guinea pig whose mother had a smooth coat. So do a Punnett square for this cross. So the rough-coated guinea pig is heterozygous, and the other guinea pig is heterozygous because the mother had a smooth coat. So when we do the Punnett square, we get 75% that would be rough-coated. That's because this individual would be rough-coated, so would that individual, and so would that individual. We have 25% smooth-coated. Heterozygous smooth-coated would be 0% because if you are heterozygous, you'd be rough-coated. So it's not possible to be heterozygous and smooth-coated. Heterozygous rough-coated would be 50%, these two individuals right here. Now remember, these Punnett squares represent probability of producing offspring with different genotypes and therefore phenotypes, but this doesn't represent actual individuals. It's a probability problem. In fruit flies, long wing is dominant over short wing. If you have two long-winged fruit flies, they both have the dominant phenotype, and they produce offspring. They produce 49 short-winged and 148 long-winged. So the genotypes of both parents would have to be heterozygous. And if you look at the ratio, 148 to 49, it's roughly the 3-to-1 ratio, which you should be familiar with by now. And if we do our Punnett square, we are crossing two heterozygous individuals, and we have big L, big L, big L, little L, big L, little L, and little L, little L. So we have one homozygous dominant, two heterozygous, and one homozygous recessive. So all of these individuals would have long wings, and these individuals would have short wings. We would have fewer of them. A man and his wife are heterozygous for brown eyes. They have 6 children who all have blue eyes. How is this possible? Well, random assortment. In every zygote, there's a 25% chance of producing a homozygous recessive individual. It's not likely to occur, but it's possible. So how can we predict the chances of them having 6 children in a row with blue eyes? Well, it would be 1/4 to the 6th power, or 1 over 4,096. The way we figure these out is to understand the laws of probability. So the first law of probability states that if you want to know the chances of 2 events occurring together, that you have to find the product of their individual probabilities. So that means you have to find the fraction of each of those chances and then multiply them. So each time they produce offspring, there is a 1/4 chance of having a blue-eyed child. And if you want to know how likely it would be for that to happen 6 times in a row, it would be 1/4 times 1/4 times 1/4 times 1/4 times 1/4 times 1/4. So what is the probability that their next child would have brown eyes versus blue eyes? When we do the Punnett square, we have a 75% chance of brown-eyed child and a 25% chance of having a blue-eyed child. The second law of probability is that the probability of one event does not affect the probability of the next event. Sometimes you'll hear people who have children and they have many boys in a row or many girls in a row, and they're kind of hoping to have at least one child of each sex. So they keep having kids, and you'll hear them say, oh, we had 4 boys in a row. We really wanted a girl. What are the chances that the next one would be a boy too? Well, it's 50-50. So now we're going to do dihybrid crosses. In pea plants, red flowers are dominant over the allele for white flowers, and tall plants are dominant over short plants. So a farmer bred 2 plants that are heterozygous for both traits. That means that the genotype of the parents would be big R, little r, big T, little t. Now, when we set up our Punnett Square, we need to find the possible combinations of alleles in gametes. Each gamete would have one R and one T because these alleles are on homologous chromosomes. And when meiosis occurs, you get one of each homologous chromosome. So the R's would separate and the T's would separate. So in order to find the possible combinations, remember you saw in the lecture video to FOIL: first, outside, inside, last. So it would be big R, big T, big R, little r, big T, and little r, little t. Then filling this in is not difficult. It can be a little bit tedious though. You keep your R's together and your T's together. It wouldn't matter if you started with T's first and then did your R's, so long as you're consistent throughout. If you notice here, people always look at this and say, well, wait a second, I thought you put the dominant alleles first. But remember, these T's are not dominant to the R's. So if you started off putting your R's first, you just keep your R's first. So what are the possibilities of getting a tall red plant? If we look here, we have all of these are tall and red and all of these are tall and red. And then there's tall and red and tall and red. And if we count them all up, it is 9 out of 16. And short and red would be 3 out of 16. And tall and white would be 3 out of 16. And short and white would be 1 out of 16. Our short and white are here. Our tall and white are here. And then we have our short and red here, here, and here. Whenever you have two parents that are heterozygous for two traits and neither trait shows incomplete dominance or codominance, you will get this 9 to 3 to 3 to 1 ratio. It only works if both parents are heterozygous, and it only works if your two traits show complete dominance. In man, free earlobes are dominant to attached earlobes, and non-red hair is dominant to red hair. A homozygous dominant man marries a homozygous recessive woman for the traits above. The genotypes of all of their children would be heterozygous for both traits, big F, big N, little n. And the children would all have free earlobes and non-red hair. We've shown you a picture here of free earlobes versus attached earlobes, just so that you know what we're talking about. You don't even really need to do a Punnett Square for this problem, but here it is. So let's do another problem. So free earlobes are still dominant to attached earlobes, and non-red hair is still dominant to red hair. A man who is heterozygous for both traits marries a woman who's homozygous recessive for both traits. The genotype of the man is big F, big N, little n. And the genotype of the woman is little f, little n, little n. Now we put the father's gametes on top. Notice there's one F and one N. And with the female, the only thing that her gametes can have are little f and little n. So it makes our Punnett Square a lot simpler. So here is our genotype, our genotypes and our genotypic ratios, and our phenotypes and phenotypic ratios. And we would call this a dihybrid cross because we are looking at two different traits. Sometimes students, instead of reducing and giving me a 4-square Punnett Square, will give me a 16-square Punnett Square. And that's fine as long as you reduce your 4 to 4 to 4 to 4 ratio and recognize that it's the same as a 1 to 1 to 1 to 1 ratio. You wouldn't lose points, you would just lose time as long as you reduce your ratio at the end. And typically, if a student forgets to reduce their ratio, but they get the correct ratio in the non-reduced form, I usually give half credit.